For the single spin in diamond, depending on how strongly it was emitting light, you could infer if it was up or down. Here, for this cryptochrome, just by looking at how strongly this guy is emitting light, this protein is emitting light, you can actually infer whether the chemical reaction continued through the singlet branch or through the triplet branch, or which combination of probabilities that was. Also, uh, although this evidence is not super absolutely unquestionable, there is evidence that shows that seems to suggest that cryptochrome coherence times at room temperature are at most on the order of 1 microsecond. Again, which is on par with the 2 microseconds that I quoted for the envy center in diamond. Okay, so this is very encouraging. The problem is that the next step of evidence for this mechanism occurs at huge length scales. Okay, occurs for experiments with birds, experiments with flies, experiments with thousands of cells in solution, and those experiments, they are consistent with the things that people see in vials of cryptochrome, but it's really hard to say, well, the bird is behaving the way it is because there's some quantumness going on in it for a short time. So before I argue that we need to bridge those length scales, let me just describe some experiments done with, say, birds. The problem with experiments with birds is that it's hard to make them. The sample size is small, but people do capture birds, migrating birds during migration season. They put them in cages with external, with shields, with Faraday cages around and coils inside. And by changing the magnetic fields that the birds see, the birds want to go out of the cage via different directions. People can also train flies. Okay, I don't know how they do this. It's harder than quantum mechanics to me. But they train flies to find food based on the presence of a magnetic field. Then the researchers knocked out, they removed the cryptochrome gene from the flies, and the flies were no longer able to find food based on magnetic fields. Then in a further experiment, the researchers put back human cryptochrome inside the flies, and the flies were back to being able to find uh, food via the presence of a magnetic field. So all those things are consistent with the microscopic model. But then again, it's very hard, right? So what we're starting to do here at UCLA, we're super delayed because of COVID and stuff, but we persist. What we want to do is to bridge those length scales. What we want to do is, if you will, quantum sensing, chemically inspired experiments on cryptochromes in vivo inside, say, single cells. What we are building are different types of glorified microscopes with coils. Okay, so what we want is to explore those spins in proteins as bona fide quantum sensors that act at room temperature and in noisy environments, right? So if spins in proteins are treated as bona fide quantum sensors, there's this huge phase space for us to take technology from the field of quantum computing, quantum sensing, spin physics, atomic physics, and start applying those techniques onto those spins in biology. And I'll tell a little bit more about this, but before I want to mention that, again, this is not a talk about birds, okay? This is more widespread than that. Right now, there is, again, evidence at large length scales that this exact same physics of spin-dependent chemical reactions might underlie many relevant biosensing phenomena, such as metabolic regulation, uh, stem cell mediated growth, in this case in planaria. There's also studies on human stem cell epigenetics that are altered by weak magnetic fields. DNA repair is altered by weak magnetic fields. This last picture that I post there, it has a story, right? And I'm going to spend my minus 2 minutes remaining to tell this story. So I have a friend in Munich. His name is Peter Fierlinger, and he's a precision measurement physicist. So for a living, he builds hypomagnetic chambers, and he puts cold atoms experiments inside to do precision measurements, and for that they need very, very low spurious magnetic field. So he can build hypomagnetic chambers with DC magnetic field levels on the nanotesla range. Again, the magnetic field of the Earth is 50 microtesla. So just for kicks, what he did, and actually another group in Mainz replicated this experiment, is the following. They took tadpoles. Again, they're physicists, but they wanted to play with tadpoles. They took tadpoles and they grew inside those those hypomagnetic chambers under two different conditions for 2 days. In one condition, they grew tadpoles just with this DC nanotesla magnetic field. And in the other condition, they grew the tadpoles inside this hypomagnetic cage with an internal applied magnetic field mimicking the 50 microtesla magnetic field of the Earth. The tadpoles grown under 50 microtesla were okay. The tadpoles grown under the absence of magnetic fields were, as you can see there, about 30% macroscopically malformed and weird looking. This is not a proof that this is spin mediated, but given the tiny magnetic field strengths involved, my best educated guess right now is that this is spin physics uh, dependent. And again, this poses all sorts of questions, right? From disease sort of stuff, like can we start tapping on those endogenous quantum mechanical degrees of freedom to drive physiological processes in ways that we need to be able to predictably know, right? Questions from that to like space exploration, right? What's the magnetic field in Mars? Can we reproduce in Mars? Can we go to grow tomatoes in Mars, right? So I think that there's a lot of uh, interactions of biological materials with tiny magnetic fields via spin physics that needs to be explored. So this field is lies within a bigger field called quantum biology, right? And I maintain that right now the bottleneck for spin mediated quantum biology is the sort of high tech instrumentation that we are going after. When we say that we're building microscopes, the microscopes don't look like the microscopes to the left of the pictures there. It looks like the thing to your right. It's like an atomic physics experiment with plenty of mirrors, electronics around that is run onto biological samples. I won't have time to talk about that, but it's very important that our experimental approach is sort of driven by mathematical models on this spin physics. What we want to do is model the spin physics and then make predictions and see where we are with this model in the lab. Even like we as crappy experimentalists, we can already do um, simplified spin simulations on what's going on. The simplest model that one needs to use if you just do a open quantum systems Liouvillean evolution simulation is like you have two electron spins that start entangled, start in a singlet state, but that otherwise do not interact among themselves. Okay, this spin is free, and this guy has hyperfine interactions with say one nuclear spin nearby. With this three spin uh, crappy simulation, you can already start making some very interesting predictions. And I promise that the last thing that I'm going to explain to you, but have a look at this top right plot here, where this is a simulation from our code. We're not the first person to people to do these simulations, but we can also do those simulations. So here we plot the magnetosensitivity of this putative of this cryptochrome using again this model plus photophysics parameters from the literature. So magnetosensitivity, that is how well the protein is at like distinguishing between the two pathways, the two chemical pathways. If the chemical reaction continues through 50% of the time through this pathway and 50% of the time through this pathway, this is a very poor, this is a field for which there's very poor magnetosensitivity, right? Because it's almost random. But if for a given magnetic field intensity, frequency, or direction, the protein responds with the chemical reaction going 95% to this branch and 5% of the times to this branch, that's a very good magnetosensitivity. So our simulations is that the magnetosensitivity of this protein, right, as a function of applied external field, it's not monotonic, it goes up and down. At 10 times the magnetic field of the Earth, the magnetosensitivity is already very, very tiny. So going for stronger fields does not help, it harms, because again, this mechanism only works as long as those two electronic spins see different local magnetic field environments. If you start applying very strong magnetic fields, the hyperfine interactions that make all the difference are going to be overpowered by this external magnetic field, and changes in this external magnetic field are not going to cause differences in Larmor frequency phases. Okay, so this goes up and down. Interestingly, the maximum of the magnetosensitivity is very close to the magnetic field of the Earth, which is either a coincidence or it might show that those proteins evolved to work under the conditions where they evolved. And we think we can distinguish between those two hypotheses by doing, we cannot do this, but people who can do directed evolution of proteins could actually distinguish between those two properties. And that's the last thing I wanted to say for what we do. The mission of our group is to unambiguously determine whether quantum physics can be established to account for relevant biological phenomena, and importantly, be manipulated to technological or therapeutic advantage. Therapeutic advantage by driving physiology, right, in ways that haven't been mapped before. And technology, we also think that we can learn with nature how to build better quantum systems, especially because if nature is using quantum mechanics to function, it knows damn well how to deal with noise. Okay? We're not the only people actually thinking about this problem. There are other people doing high-tech experiments on biological systems, and I do think that interdisciplinarity is what's going to drive cool things in science forward in this century. So we've made the turn. I have to like take out my hat to the people in the group. There are chromophores in cells that are not exposed to light, right? There's a lot of photo-driven, potentially photo-driven things in cells that are, say, in your liver, right? That do not see light. So I think that in redox reactions, flavoproteins in general, and I think this is super more widespread than thought. There are crazy papers from the 60s from Russian scientists. So Russian scientists are really cool because I think that Russia did not develop this stigma of magnetic fields, electric fields in biology that I think is present in the West. So I think those people continued with this research for a long time. So there's papers that say that phosphorylation processes are magnetic field dependent. There are like books on what has been tried and what has been seen by applying weak magnetic fields and what things change. And people see all sorts of weird changes in gene expression. I think it's super widespread. And again, this is not data driven. This is my best guess that movement of ions inside cells when transient, right? But that's fine because our quantum sensors are transient too. Movements of ions inside cells when they're viewed from very close by, those might be generating strong enough magnetic fields to shift this balance of like those chemical dependent spin dependent chemical reactions. So I think it's in many biological processes actually. Also spin transport, but I won't have time to talk about it.